Welcome, everyone. Thank you for joining us. People are still logging in. We will get started momentarily. Welcome to everyone who has just logged in. People are still connecting, and we will get started with our presentation momentarily. Welcome, everyone. People are still logging into our program today, and we will get started in just a few moments. Okay, good afternoon, everyone. My name is Avraham Grohl, and I'm the Executive Director of JewishGen.org. Welcome to this week's Jewish Gen Talk with Dr. Steve Morse, who will be speaking on the topic of From DNA to Genetic Genealogy, everything you wanted to know, but were afraid to ask. We are glad you are with us. A couple of notes before we get started. First, this presentation will last for about an hour, followed by a few minutes of questions. You can post your questions in the QA box, which is accessible on the bottom of the screen. Following the presentation, we will be distributing a 29-page guide that Dr. Morse has created. Finally, I'm pleased that we once again have a very large crowd of approximately 3,000 people who are participating on this webinar and joining us from around the world. And in this regard, I would like to thank Nancy Siegel, our Director of Communications, and Richie Baum, our Communications Editor, for all of their hard work in making this Jewish Gen Talks series so successful. We have one more talk after this week, then we'll take a break for the next month. The talk will be given by Dr. Sally-Ann Sack. She will be speaking about Jewish names, and you can register for her talk by visiting JewishGen.org/live. Dr. Steve Morse is someone who is well known in both the genealogy world and the technology world. He is the creator of the award-winning One Step website. He is the recipient of the Lifetime Achievement Award from the International Association of Jewish Genealogical Societies and the Award of Merit from the National Genealogical Society. And he's best known perhaps as the architect of the Intel 8086 chip, which sparked the PC revolution 35 years ago. The topic today that Dr. Morse will be speaking about is timely not just for its genealogical application, but for the lessons it teaches us. The Talmud emphasizes that equality should exist among all people on earth, since ultimately we trace our DNA and our roots back to the same two people, Adam and Eve. And it is for this reason that the world was not created fully populated, so that no one could say that his or her parents were superior to their neighbors. So within this context, and without further ado, Dr. Steve Morse.
Thank you, Avrami, for that very nice introduction. I should note that this lecture will have the narration embedded within the lecture, so I'll just be sitting here practically doing nothing. But what I will do, I will working my mouse, so I'll be able to point to different parts on the screen. And also, since the audience, since you can't see or hear the other members of the audience, I realize that puts you at a disadvantage. You don't know when to laugh and when to applaud. So I'll be standing here with signs holding them up at appropriate times. Okay, with that, let's get started. I will start the thing running right now.
My name is Steve Morse, and today I'll be presenting a lecture on genetic genealogy and DNA. You are probably wondering why I'm giving this lecture and what are my qualifications for doing so? Well, I have no qualifications. I'm not a geneticist, not a biologist, and not a chemist. And I have no affiliations with a DNA testing laboratory. I'm an engineer. I'm giving this lecture because DNA has become a hot topic in genealogy. Unfortunately, most of the DNA lectures being presented assume that the listener knows the basics. I didn't have that knowledge because DNA was invented after I went to school. I knew a little about genes having studied Mendel's laws in high school biology. and I remembered that chromosomes determine sex. But beyond that, I was lost. I didn't even know how genes related to chromosomes. So I decided to teach myself about genes, chromosomes, and DNA. While doing so, I realized that I was not alone. Most genealogists were as confused as I was. So I developed this lecture based on what I learned and how I learned it. It is my hope to give you the background you'll need to be able to understand this relatively new field. The lawyers made me put up this next slide. But perhaps this is a good time for me to tell you what this talk is about and what it is not about. I don't want anybody to have false expectations. There are three basic DNA tests that genealogists are interested in. One is a test that will allow you to trace your direct paternal line. Another allows you to trace your direct maternal line. And the third test lets you trace all lines. The direct maternal and direct paternal tests are based on science and are the most accurate. The third test is based on statistics. For years I dismissed that third test as not having much validity. But more recently I've changed my mind as I've seen some amazing results with my own matches. This talk will cover the direct paternal and direct maternal tests. The three major testing laboratories used to give all three tests, but today all but one have stopped doing the direct paternal and maternal tests. This talk will not teach you how to compare your matches to see who you are related to, but it will give you an understanding of what the matches mean and why you might be related to somebody who was a close match. The field of genetics originated in 1865 with Gregor Mendel and his pea experiments. 165 years later, it evolved to the point where it could be useful to genealogists, as exemplified by Megan Smoleniak's coining of the term genetealogy in the year 2000. It was a catchy phrase combining genetics and genealogy, but unfortunately it never caught on. Everything we need to know about genetealogy involves three basic concepts: genes, chromosomes, and DNA. When I started teaching myself this topic, I didn't know how they were related, nor that they were even related. So let me jump ahead and tell you the answer. The relation between them is simple. Traits are determined by genes. Genes are located on chromosomes. Chromosomes are composed of DNA. Now let's look at each of them in more detail. Mendel In particular, he concluded that each trait is determined by a pair of discrete entities or genes. One gene of each pair comes from each parent, Mendel is credited as being the father of genetics, but he never actually saw a gene, nor did he ever use that term. Instead, he postulated the existence of discrete entities and drew conclusions about them based on statistical observations from experiments involving the breeding of pea plants. In particular, he concluded that each trait is determined by a pair of discrete entities or genes. One gene of each pair comes from each parent, and the two genes are not blended. Instead, one of them dominates. So if one parent has light eyes and the other dark eyes, the offspring will not have medium-colored eyes. As an example, let's consider the color of the pea plant's flower. The flower can be either purple or white. The color is determined by a pair of genes, each of which specifies either purple or white. The purple gene is dominant. If the child pea inherits a purple gene from each parent pea, the child's flower would be purple. If the child inherited a white gene from each parent, the child's flower would be white. If the child inherited a purple gene from one parent and a white gene from the other, the child's flower would be purple because purple is dominant. And it would not matter which parent contributed the purple gene and which contributed the white gene. So much for genes. Next, we will look at chromosomes. In the nucleus of nearly every human cell is a set of 46 chromosomes. These chromosomes are numbered in pairs from 1 to 22 for a total of 44. Each of the two remaining chromosomes are either X or Y. The numbered chromosome pairs, 1 to 22, are referred to as autosomes. The X and Y chromosomes are called sex chromosomes. The X and Y chromosomes are called the sex chromosomes for an obvious reason. They determine the sex. Males have one Y chromosome and one X chromosome. Females have two X chromosomes. One sex chromosome comes from each parent. Now a question. Since a male child has one Y chromosome, which parent did he get it from? His mother has only X chromosomes, so she couldn't have contributed it. That means the child had to get his Y chromosome from his father. Furthermore, he got his X chromosome from his mother. More about that later. That takes care of genes and chromosomes. Now for DNA. A chromosome is a long deoxyribonucleic acid molecule, better known as DNA. The DNA structure is a double helix with the two strands running in opposite directions as shown in the figure. The direction of each strand is determined by its chemical composition, but we won't say any more about the direction since it doesn't affect the genealogy story. Each of the strands consists of 4 repeating compounds, which are called bases or nucleotides. The 4 bases are adenine, cytosine, guanine, and thymine. Fortunately, we don't have to remember these names, but can refer to them simply by their initial, A, C, G, and T. The bases and the two strands always pair up in a specific way. In particular, A on one strand always pairs with T on the other, and C always pairs with G. Why are there no other pairings, such as A with G? That's due to the geometry of the compounds. Their shape is such that the only pairings possible are A with T and G with C. The other pairings simply do not fit together. It's like the pieces in a jigsaw puzzle. Let's summarize and relate the genes, the chromosomes, and DNA. Chromosomes are composed of DNA. If we unwind the chromosome, we get a long strand of DNA. A gene is a subset of the DNA sequence having an identifiable function. By a DNA sequence, I mean those A's, G's, C's, and T's. The DNA in between the genes is called junk DNA. Going down the DNA sequence of a chromosome, we encounter gene, junk DNA, gene, junk DNA, etc. In between each gene is junk. Junk DNA. That's an interesting concept. What is it? It's a DNA sequence for which no function has yet been identified. Perhaps it contains driftwoods from our evolutionary past, such as for our fur color or tail length. One theory is that it contains switches that turn the genes on and off.
For example, the gene for eye color has no function in the heart cells, so perhaps the junk DNA turns off the eye color gene in that case. Perhaps we will discover a function for it in the future, so the term junk DNA has fallen out of favor and been replaced by the term non-coding DNA. Let's look at some numbers. The actual number of genes or base pairs is not important for genealogy. But as an engineer, I like to know if we are talking about 2 things or 2 trillion things. So here are the numbers, just to put things in perspective. How many base pairs are there in a gene? The average length of a gene is 27,000 base pairs. The largest known gene has about 2.5 million base pairs. How many base pairs are there in a chromosome? That depends on the chromosome number, as shown in the chart. There are between 50 million and 250 million base pairs per chromosome. They are in size place. So the chromosome numbering is not arbitrary. They are numbered by decreasing number of base pairs. How many base pairs in all 46 chromosomes? We add up all these numbers and multiply by 2 because there are 2 of each chromosome. That gives about 3 billion. So we can completely identify a person by giving a sequence of 3 billion base pairs. No 2 people would ever have the same 3 billion base pair sequence. Even identical twins will not be identical for all 3 billion. Now let's count genes. How many genes are there in a chromosome? That depends on the chromosome number as shown in the chart. It's somewhere between 200 and 3,000. How many genes in all 46 chromosomes? When I first put this talk together, it was thought to be 30,000. Later that number was revised to be closer to 20,000. Let's consider eye color, since we all probably recall from high school biology that genes determine eye color. Specifically, we were taught that brown eyes are dominant over blue eyes, so a person would have to have 2 blue-eyed genes in order to have blue eyes. What we weren't taught, because it wasn't known at that time, is where these genes are and what base values they have. We now know the rest of the story. In the case of eye color, the controlling gene is the OCA2 gene. That gene is located on the long half, called Q, of chromosome 15 in band 1, subband 1. As far as our genealogy goes, it's not important that we know all these details. But it's fascinating to realize that geneticists have mapped out all the chromosomes and know not only where a particular gene is, but also the values of all the base pairs in that gene. In our OCA2 gene, there are about 350,000 base pairs. But only 3 of them are relevant for eye color. The location of these 3 are shown in the chart, along with the values they would have for a blue-eyed person and the values for a brown-eyed person. The DNA code is not important as far as our genealogy is concerned. It is important for understanding genetic diseases, which some genealogists are concerned with. If you're interested only in knowing who might be your second cousin, you can go to sleep now. I'll wake you up when you need to start paying attention again. However, this is the part of DNA that I find the most fascinating. I can't help but see the similarity between the DNA code and a computer program. To realize that each cell in our body contains a computer program for building our body is truly amazing. Recall that I'm not a biologist, so some of these points I'm accepting on faith. For example, I really don't know much about proteins. But the biologists tell us that every function in a living cell depends on proteins. So I will take that as gospel and realize that if I knew the rules for making proteins, I would understand all the functions in our body. Geneticists discovered that each gene is an instruction manual for making one protein.
and the instruction manual is written in the language of DNA. To understand how to read the manual, we have to know what proteins are composed of. The biologists tell us that each protein is a sequence of amino acids. Okay, I can accept that even though I'm not sure what an amino acid is. The biologists then tell us that there are only 20 amino acids as shown in the chart. The amino acids have names like asparagine, isoleucine, and my all-time favorite, phenylalanine. These are names that only a biologist could love. If they were named by a computer scientist, they would probably be called amino acid 1.0, amino acid 1.1, etc. The biologists realized that we were not going to like these names, so they gave us a 3-letter abbreviation for each amino acid. And in case we still found it cryptic, they gave us a 1-letter abbreviation as well. Of course, it's kind of confusing to see that the 1-letter abbreviation for lysine is K, but if we stick with the abbreviations and forget the names, we'll be okay. Since a protein is a sequence of amino acids, if we put several amino acids together in a particular order, we will make a particular protein. For example, here are the chemical structures for 3 of the amino acids. And here is a protein made from a sequence of these amino acids. If we rearrange the amino acids, we would have a different protein. And if we use different amino acids, we would have a different protein. Now we are ready to talk about the DNA code. Recall that the DNA code in each of our cells consists of a sequence of about 3 billion base pairs. What was discovered is that within the gene, 3 consecutive base pairs, called a codon, specifies one amino acid. This is shown in the diagram. Note that the DNA sequence starts off with the codon GCA, and that specifies amino acid A. Don't confuse amino acid A with base pair A. They were 2 different things. The next codon is AGA, specifying amino acid R. Then comes GAT, specifying D. And AAT specifying N. An entire gene specifies a sequence of amino acids, which is a protein. So we need to know the amino acid associated with every possible sequence of 3 base pairs. The biologists have figured it out, and the result is summarized in this chart. Note there can be more than one codon specifying the same amino acid. For example, codon AGT and codon AGC both specify the amino acid S. Note also that there are 4 special entries in the chart. 3 of them, TAA, TAG, and TGA, do not specify any amino acids, but rather serve as a stop code. They are saying that you have reached the end of a gene and there are no more amino acids in the protein. The 4th, ATG, denotes the amino acid M, and also serves as a start code saying that you are at the beginning of a gene. This, of course, implies that all proteins start with the amino acid M. Here is an example of a DNA sequence for making a protein. We read this by going through the sequence looking for a start codon, ATG. When we find that, we know we are at the start of a gene, and the first amino acid is M. Everything preceding the ATG is junk DNA, I mean non-coding DNA. Next, we see the codon CTG, and that specifies the amino acid L. We continue scanning the bases 3 at a time until we get to a stop codon. In this case, the stop codon we get to is TGA. That tells us that we have reached the end of the gene and there are no more amino acids for this protein. The bases thereafter are all junk DNA.
Okay, those of you who went to sleep can wake up now. We're going to be talking about genealogy again. Specifically, I'm going to tell you about the birds and the bees, or how we inherit chromosomes. Usually, when we talk of inheritance, we are talking about what is passed from parent to child when the parent dies. This is not what we would be talking about here. In genetics, inheritance refers to what is passed from parent to child when the child is born. We will look at two kinds of inheritance, autosomal inheritance and sex chromosome inheritance. And we will learn how both of these can be used to tell us something about our ancestors and therefore our genealogy. And in case you've already forgotten, autosomes refers to the numbered chromosomes 1 to 22. We'll first look at autosomal inheritance. And again, recall the autosomes are the numbered chromosome pairs 1 to 22. Let's consider chromosome number 7 as an example. The same story applies to all the other autosomes, but I just like picking on number 7. It's my favorite scapegoat. By focusing on just one chromosome pair, the story is easier to follow. Since a child inherits one number 7 chromosome from each parent, and since each parent has two number 7 chromosomes, it would be natural to think that the father passed one of his number 7 chromosomes directly to the child, and the mother did the same. This is depicted in the figure. But if this were the case, then the number 7 chromosome that the father passed to the child would have come from one of his parents, say his mother. And the child's mother would have gotten the number 7 chromosome that she passed from one of her parents, say her father. So the child would have genetic information on number 7 from his paternal grandmother and his maternal grandfather. He would have nothing from his other two grandparents. The genes on those grandparents' number 7 chromosomes will be lost to all future generations descending from this child. However, that is not the case. Instead of the father passing one of his number 7 chromosomes intact, his two number 7 chromosomes are shuffled together, forming a new chromosome that gets passed down. The mother does the same. So the child receives one number 7 chromosome from each parent, but each of those chromosomes is a mixture of the parent's two number 7 chromosomes. By this means, genetic information on all the autosomes from all the grandparents gets passed down. This is shown in the revised figure. Now for sex chromosome inheritance. Previously, we said that every cell in the body contains 46 chromosomes, specifically 22 autosome pairs and 2 sex chromosomes. That's not completely true. The exceptions are the sperm cells and the egg cells, which are the seeds for the next generation. These cells have only 23 chromosomes, made up of 22 unpaired autosomes and 1 sex chromosome. To see how the sex chromosomes are inherited, let's look separately as to how baby boys are made and how baby girls are made. Recall that boys have 1 Y and 1 X chromosome, whereas girls have 2 X chromosomes. When a boy is made, the father produces a sperm cell with a Y chromosome. That Y sperm cell fertilizes an X egg cell from the mother, resulting in the offspring having 1 X and 1 Y. The X in the mother's egg is a shuffling of the mother's 2 X chromosomes, but the Y in the father's sperm is not the result of a shuffling. Instead, it is the father's Y chromosome intact because the father doesn't have another Y chromosome to shuffle it with. When a girl is made, the father produces a sperm cell with an X chromosome. That X sperm cell fertilizes an X egg cell from the mother, resulting in the offspring having 2 X chromosomes. As in the boy case, the X in the mother's egg is a shuffling of the mother's 2 X chromosomes. And again, the father's sperm is not the result of a shuffling. Instead, it is the father's X chromosome intact because the father doesn't have another X to shuffle it with. Note that in both cases, the X chromosome that the child receives from the mother is a shuffled X chromosome. But the Y chromosome that the son receives from the father is an intact Y chromosome since the father had only 1. Similarly, the X chromosome that the daughter receives from the father is an intact X chromosome. Although the daughter received the father's X chromosome intact, that chromosome doesn't stay intact for the next generation. When that daughter has children, her two X chromosomes are shuffled together, so her father's intact X chromosome is no longer intact. That is not the case for sons. The father's Y chromosome is passed from father to son to grandson to great-grandson, and so on ad infinitum, always intact. Now we can answer the question of who's your daddy. Also, we'll see why mutations are a good thing as far as genealogists are concerned. We can use our knowledge of chromosome inheritance to determine if two people share a recent common ancestor and are therefore not too distant cousins. We can also use it to determine something about our early ancestors who migrated from someplace in Africa and can determine if our ancestor turned left or turned right when he got to Kenya. Such distant ancestry won't help us instruct our family tree, but some people like to know these things. Like father, like son, or the apple doesn't fall far from the tree. The key to doing this sort of genetic testing lies in the way the sex chromosome is inherited. As already noted, the Y chromosome is never shuffled. It is always passed from the father to the son intact. So the DNA code on the son's Y chromosome should be identical to the father's, and every male should have the identical Y chromosome to Adam. In that case, we could do DNA testing and determine that we are all descendants of Adam. Although this might help us weed out the extraterrestrial aliens among us, it would be of little help in constructing our family tree. But fortunately, mistakes happen, and the DNA is not always passed intact. Such mistakes are called mutations. Considering the vast number of cells in our body, in excess of 10 trillion, mutations are probably happening all the time. But if any one cell mutates, it probably never gets noticed. The exception is in the sex cells, the egg and the sperm, because such mutations will get passed down from parent to child. So thanks to mutations, the Y chromosome passed from father to son will sometimes change, and the son's Y chromosome might differ slightly from the father's Y chromosome. It is this difference that will allow us to find out if two people are related and how close the relation is. There are two kinds of mutations that we are most interested in. The first is referred to as a SNP. SNP is an acronym for single nucleotide polymorphism. Those are big words, but the important word is single. It refers to a mutation in which a single base pair changes its value. Recall that nucleotide is another name for base. The second mutation is a STIR. STIR is an acronym for short tandem repeat. Again, we can ignore all these words except for one, repeat. It refers to a DNA sequence that repeats itself several times. Due to a mutation, the number of times it repeats could change either upward or downward. SNP, single nucleotide polymorphism. As an example, a DNA sequence of TGAT at a particular location called a marker might become TGCT when passed to the next generation. A SNP is an extremely rare event. It is so rare that if it occurs at a particular marker to any person at any time in history, the probability of it ever occurring again at the same marker to any other person would be zero. That is, if the mutation occurred to one man who lived 75,000 years ago, no other man in all of history would ever mutate at that same marker. And for the same reason, once the mutation occurs and gets passed down to future generations, it will never get undone. Where in the DNA sequence shall we test for SNPs? What would happen if it occurs inside the DNA sequence of a gene? This could have a profound effect on the organism, and the organism might not even be viable. So it would make no sense to look for mutations in genes in order to find our ancestors. People having such mutations probably won't live long enough to have descendants. What if it occurred outside a gene, in so-called junk DNA?
In this case, it will have no effect whatsoever. For this reason, genealogists are interested in testing for Snips in junk DNA only. Because it is such a rare event, it can be used to trace early migration patterns. If you and I both have the same SNP value at a particular marker, then we must both be descended from that man in which the SNP occurred 75,000 years ago. This is not going to tell us if we are second cousins or not, but it will tell us if we descended from the tribe in Africa that turned left when it got to Kenya. Stir, short tandem repeat. As an example, a DNA sequence of CTA repeated 5 times suddenly gets repeated 6 times. Usually involves 2 to 10 base pairs. Unlike SNPs, stir mutations are not at all uncommon. They happen on average once every 500 events. That means if 2 people have the same stir value at a particular marker, they probably have a common ancestor within the last 500 generations. Well, that means they are cousins but quite distant. If they have the same stir values on more than one marker, we can reduce that number considerably and might be able to deduce that their common ancestor was only a few generations back. So the stir values can be used to find if 2 people are related within the time frame of interest to genealogists. The change in repetitions can be an increase or a decrease. For the same reason as SNPs, the stirs tested to determine relationships are in the junk DNA regions and not in the genes themselves. As already mentioned, this can be used to estimate the time to a recent common ancestor. So who's your mommy? Up till now, we have examined the direct male line based on the Y chromosome. Something similar can be done with the direct female line. Yes, girls can trace their genealogy too. The 46 chromosomes are in the nucleus of each cell. Surrounding the nucleus are the energy bars of the cell, the mitochondria. Like the chromosomes, the mitochondria are composed of DNA. It's called mitochondrial DNA or simply mtDNA. The sex cells, egg and sperm, are no exception, and they too contain mitochondria. So it would seem that a child should get mitochondria from both parents. However, the mitochondria is in the tail of the sperm cell. The sperm that actually penetrates the egg works so hard to do so, and it barely manages to get its head in. The tail breaks off and never makes it. So the child gets its mitochondria from the mother only. The mitochondria is passed from a mother to all her children, but only her daughters pass that mitochondria to their children. Her son's children will get their mitochondria from their mother. That means that every woman received her mitochondria from her mother, who in turn received it from her mother, and so forth. This allows us to trace female lineage. If not for mutations, all women would have mtDNA identical to Eve's, but mistakes do happen, and mtDNA mutates. The mutations are of the SNP type and occur very infrequently, so they can be used for tracing early migrations. Did your mother turn left or right when she got to Kenya? They are not that useful for determining if 2 women have a common great-grandmother. Out of Africa, the road we traveled. I'm going to describe early migration patterns, and I'll do it in a story format. As such, I will take poetic license. I'll embellish and I'll improvise, but the facts presented are all true. In the beginning, God created man. Well, that's neither a fact nor an embellishment. It's based on religious belief. And all men lived in Africa and had identical Y chromosomes. Fact. And in the year 60,000 BP, before the present, a mutation occurred. Fact. One man was born with a different base value at marker M91. Fact, a SNP mutation. And he was shunned by the others and forced to leave the tribe. Okay, I'm embellishing to make a point. So he started his own tribe and called it Haplogroup A, fact. His tribe remained in Africa, primarily around Ethiopia, fact. And then another man in the original tribe was born with a mutation, and he too was asked to leave, embellishment. He started the BR tribe, fact. Then, in the year 50,000 BP, 2 mutants appeared in the BR tribe. The first started the B tribe and became the Pygmies. The second started the CR tribe and immediately had 2 mutant sons, and they started tribe C and DE. The C tribe left Africa and went to Australia and the surrounding areas, Japan, Siberia, North America. Fact, 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 and fact. And so this continued, and the tribes D through R appeared, and each tribe went off in a different direction to populate the earth. This chart shows the migration paths taken by the various groups. How do we know all this? By obtaining DNA from various indigenous groups around the world and looking at their SNP values. The preceding migration pattern is based on Y chromosome mutation and tells us about paternal lineage. Similar results can be obtained from maternal lineage based on mtDNA mutations, as shown in this chart. Now let's look at some examples of DNA testing. First is the Anastasia mystery. This is a heart-wrenching story, and once you hear it, I'm sure you'll agree. It's easy to see why it was made into a motion picture with top-rated stars, and also into a full-length animated movie. The Romanov family ruled Russia for over 300 years. The Romanovs were overthrown in the Russian Revolution of 1917, and the royal family was placed under house arrest. Lenin realized that there could be a counter-revolution to restore the original bloodline, so in 1918, Lenin ordered the Red Guard to kill the royal family. Those murdered were the Tsar Nicholas II, his wife, the Tsarina Alexandra, his 4 daughters, Olga, Tatiana, Maria, and Anastasia, his son Alexei, a physician who had the misfortune of being present, and 3 female servants. I mention this because knowing who was murdered will serve as a clue for solving the mystery. The location of the burial site was never disclosed. That was in 1918. In 1920, a woman was found in Berlin, claimed amnesia, and took the name of Anna Anderson. In 1922, Anna Anderson revealed that she was the youngest daughter, Anastasia, and that she escaped the massacre. Her critics were skeptical and claimed she was Franziska Szankowska, a Polish factory worker. Well, everybody has critics. When Anna Anderson died in 1984, her true identity was still unproven. In 1991, a burial site was discovered in the Ural Mountains, not far from where the massacre allegedly took place. 9 bodies were exhumed from the site. They consisted of 2 adult males, 4 adult females, and 3 younger females. Testing of nuclear DNA, i.e. the chromosomes, revealed that one of the adult males was closely related to the 3 younger females, and that one of the adult females was closely related to the 3 younger females. The conclusion was that they were a family group consisting of a father, a mother, and 3 daughters. So could the 9 bodies be the Tsar, Tsarina, 3 daughters, the physician, and the 3 female servants? And, if so, what happened to the 4th daughter and to the son? It's time to look at family trees. We'll start with a tree of the British royal family, the House of Windsor. Here is the tree from Queen Victoria down to the present-day Queen Elizabeth. But why are we looking at the British royal family when we're trying to solve a mystery with the Russian royal family? That's because Queen Victoria had another child, Alice, who was the mother of the Tsarina Alexandra. Here's the British family tree that includes the Romanov family. To solve the mystery, DNA was tested from Prince Philip, husband to Queen Elizabeth. But why Philip? He married into the royal family, so why would we expect him to have any of the royal DNA? That's because Alice had another daughter, Victoria, who was the grandmother of Philip. So Philip and Elizabeth were both descendants from Victoria in their own way, and were related, third cousins, before they became husband and wife. Now it's time to follow the mitochondria. Recall that mtDNA is passed from mother to all her children, but only her female children pass it to their children. This tree shows all people who have Victoria's mtDNA. From this, it is clear that Philip would have the same mtDNA as the Tsarina and all her children. And he would be the only descendant of Victoria alive today who has that mtDNA. So Philip was tested to determine if his mtDNA matched that of the exhumed family. Recall, however, that mtDNA mutates very slowly, and for that reason is usually not used to determine recent ancestors. Prince Philip's mtDNA exhibited a rare mutation at two specific markers. The exhumed family had the same rare mutation. This proved that the exhumed bodies are the Romanovs. An mtDNA match on the Tsar side makes the case stronger yet. The mtDNA of the exhumed body of the alleged Tsar contained an even rarer anomaly, namely two DNA values at one marker, called a heteroplasmy. Since the location of the Tsar's brother's grave was known, he was exhumed and tested. It contained the same heteroplasmy. There was now no question that the remains were that of the royal family, and the missing son and daughter gave credence to Anna Anderson's claim of being Anastasia. The only way to solve the Anastasia mystery now was to test Anna Anderson's DNA. But Anderson had died 7 years before the gravesite had been discovered. Fortunately, her DNA had been preserved. One report I read said that it came from a biopsy sample wrapped in paraffin and stored in a hospital freezer. Another said it was from a lock of her hair that her husband had saved. From whichever source, we now had a sample of her mtDNA for comparison purposes, and the results came back very conclusively. It did not match. Anderson was a fraud. However, Anderson's mtDNA did match that of Karl Macher, maternal great-nephew of Polish factory worker Franciszka Szankowska. But what did happen to Anastasia and Alexei? Would another Anna Anderson come forward in the future to claim the title? In 2007, another burial site was discovered close to the first one. It contained the bodies of a young boy and a young girl. DNA tests revealed that they were from the same family as the bodies at the other site, so they must be Anastasia and Alexei. Mystery solved. Well, not completely. Now that they had the bodies of all the daughters, they were able to put them in age order, and they discovered that the newly found body was that of Maria and not Anastasia. They had Anastasia's body all along, but because of Anna Anderson, they had assumed that Anastasia was the missing one. And note that they were fortunate in being able to solve this with mtDNA, in spite of the fact that mtDNA is more suited for determining distant ancestry. In this case, mtDNA was all that they had, because it usually is all that survives in skeletal remains. Next example is the legacy of Genghis Khan. Genghis Khan and the Mongolian Empire. 800 years ago, Genghis Khan conquered a region from the Pacific Ocean to the Caspian Sea. That's an incredibly large area, starting at the eastern edge of Asia and going halfway through Europe. His armies massacred the males and raped the females in the conquered lands. And Khan himself always got first pick of the females. Khan's eldest son had 40 sons. His grandson had 22 legitimate sons, and he added 20 virgins a year to his harem. Harems and concubines were the norm, and the males were very prolific. All told, this made an excellent petri dish in which to spread Khan's Y chromosome. Fast forward 800 years. Today, 1 in 12 men, or about 16 million men, in the region of the former Mongolian Empire carry a common Y chromosome. Furthermore, it has been established that this lineage of Y chromosome started about 1,000 years ago. And it is found in only one population outside of the Mongolian Empire, namely Pakistan, where they have an oral tradition of being descended from Khan. The conclusion is that we have isolated Khan's Y chromosome, although the original mutation predates him by several generations. So it probably started with his great-great-great-grandfather or thereabouts. Of course, we'll never know for sure if this is Khan's Y chromosome unless we find his body and test it, but the evidence is overwhelming. And the third example is the Thomas Jefferson affair. In 1802, James Callender, disgruntled because Jefferson wouldn't appoint him as Postmaster General, alleged that Jefferson fathered several children with Jefferson's slave, Sally Hemings. Unlike Clinton or Trump, Jefferson's policy was to offer no public response. However, Jefferson's daughter Martha denied the report. Keep in mind that there were 3 Marthas in Jefferson's family: his wife Martha, his sister Martha, and his daughter Martha. And 2 of daughter Martha's children maintain that Jefferson's nephews from his sister Martha, namely Peter and Samuel Carr, were the father. Let's look at the Jefferson family tree and see if Y chromosome testing will help here. Thomas himself had only daughters, except for a son who died before he was even named, so we won't be looking for any of Thomas's direct male descendants. Thomas had a brother Randolph, but they were unable to locate any living direct male descendants of Randolph. So we go back another generation to Thomas's uncle Field Jefferson. They did find direct male descendants of Field, and testing them will give us the Jefferson Y chromosome. Nephews Peter and Samuel Carr, who was implicated as possibly being the father of Sally's children, do not share a common male ancestor with Thomas, so they would have a different Y chromosome. There are no known direct male descendants of Peter or Samuel alive today, nor are there any known living direct male descendants of their father Dabney Carr. But there are living direct male descendants of Dabney's father John Carr. So now we have the Carr Y chromosome. Now let's look at Sally Hemings' family tree. Now let's look at Sally Hemings' family tree. Note that Thomas Jefferson himself is actually in Sally's tree because Sally and Jefferson's wife Martha were half-sisters. An interesting fact, but totally irrelevant to our DNA testing. Sally had 6 documented children. A 7th person, Thomas Woodson, has not been documented as being an offspring of Sally's. But oral tradition amongst Woodson's descendants state that he was parented by Thomas and Sally. They were able to locate direct male descendants of Thomas Woodson, so they have his Y chromosome. And they were able to locate direct male descendants of Sally's son Thomas Eston, so they have the Hemings Y chromosome. By the 1990s, DNA testing had matured to the point that it could be used to unravel the 200-year-old mystery. And in 1998, a DNA test was performed. DNA tests were done on direct male descendants of Field Jefferson, TJ's uncle, John Carr, grandfather of Peter and Samuel, Thomas Woodson, alleged first child of Sally, and Eston Hemings, documented youngest child of Sally. The result was that Jefferson's Y chromosome, as obtained from descendants of Field, were found in descendants of Eston. Okay, there's the smoking gun. There was some hanky-panky, and now we need to find out who was responsible. Carr's Y chromosome was not found in descendants of Eston,
So the cars have been vindicated and they can hold their heads up high. Of course, it's too late because they have been dead for 200 years. Jefferson's Y chromosome was not found in the descendants of Woodson. That proves that Woodson was not fathered by Jefferson. He might still have been a child of Sally's from a different father, but since the oral tradition says that he was the child of Sally and Thomas, the allegation that he is even Sally's child is now in question. The researchers who performed this DNA study concluded that most probably Thomas Jefferson fathered Eston. They didn't state it as a certainty, nor did they say anything about Sally's other children. Another study was done in the year 2000. This was done by the Thomas Jefferson Memorial Foundation, which is the organization that runs Monticello. They looked at a broad body of evidence beyond the DNA. For example, Jefferson, who traveled frequently, was always in residence when each of Sally's 6 children were conceived. Also, there was a strong resemblance between some of Sally's descendants and the Jeffersons. And all of Sally's children were conceived after Jefferson's wife died. This study concluded that it is very unlikely that any Jefferson other than Thomas fathered Hemming's 6 documented children. Another study was done, this time in the year 2001. This was done by the hastily formed Thomas Jefferson Heritage Society. It used the same data as was used by the Thomas Jefferson Memorial Foundation in the 2000 study, but it reached a different conclusion. It stated that the relation between Thomas Jefferson and Sally Hemings was by no means proven. Furthermore, it suggested that the most likely alternative was Thomas's brother Randolph. Remember him? Randolph's name was never mentioned throughout history as being a candidate because he wasn't living at Monticello and he didn't have opportunity. But he had the right chromosome, so let's throw his name into the pot. In 2001 again, and another study is done. This time, the well-respected National Genealogical Society, NGS, published a report in their quarterly journal. The report said that the link between Jefferson and Hemmings was credible. They said it was consistent with the weight of the evidence. Furthermore, they went on to criticize the Thomas Jefferson Heritage Society's report for weakness in approach, bias toward data, and ignoring the weight of evidence. Okay, now you have it. Disagreement among the studies. So I'll let you draw your own conclusion. Genetic diseases: Down syndrome, sickle cell anemia, Tay-Sachs disease, hemophilia. Although there are many genetic diseases, these 4 have been singled out for discussion here, not because they are the most important. They are not, but because they are each well known and they each illustrate a different genetic point. Down syndrome. This disease is found in all populations and doesn't favor any particular one. It is a chromosomal disorder rather than a genetic one and involves an extra copy of chromosome 21. 2 copies of the chromosome are obtained from one parent, usually the mother, and one from the other parent. Since it is usually the mother who gives the extra chromosome, it is not surprising that a characteristic of the mother, her age, might be a factor. Sickle cell anemia. This disease is most prevalent in sub-Saharan African populations. It is due to a mutation in the beta-globin gene on chromosome 11. The mutant gene is recessive, meaning that a person needs to have 2 mutant genes in order to have the disease. The particular mutation is a SNP, whereby a GAG sequence becomes a GTG. Since the disease is so deadly, it would seem that it would eventually become eradicated simply by survival of the fittest. That would be the case except for another factor involved here, namely, having a single mutant gene gives protection against malaria. So in regions where malaria is prevalent, those who have two good genes have a better chance of dying from malaria, leaving those with at least one mutant gene alive and thriving. This is referred to as a heterozygote advantage, a fancy term meaning that it is better to be a carrier of the disease than not. Tay-Sachs disease. This disease is prevalent in three distinct populations: Ashkenazi Jews, Louisiana Cajuns, and French Canadians. This disease is linked to the hexA gene on chromosome 15, and it is recessive. Any mutation that causes the hexA gene to produce a different protein will result in the disease. There are over 90 different identified mutations of the hexA gene. Some are Snips, some are STIRs, and there are other types of mutations as well. The particular mutation that causes the disease in the Ashkenazi Jewish population is a STIR mutation, resulting in an extra repeat of TATC. Since every 3 base pairs defines a particular amino acid, inserting 4 extra base pairs changes not just the current amino acid, but alters all the following amino acids in the sequence as well. The French-Canadian mutation is a so-called long sequence deletion. So there is no relation between the Ashkenazi Jews having the disease and the French Canadians having it. The original mutations occurred in 2 different people. What about the Louisiana Cajuns? You would think that they would have inherited from the same original person as the French Canadians. After all, both populations have a French connection. But surprisingly, the Louisiana Cajun mutation is the same as the Ashkenazi Jewish one. Makes you wonder if perhaps there was a Jewish fur trader down in New Orleans. Hemophilia, more prevalent among men than among women. It is caused by a mutant F8 or F9 gene, there are 2 types of hemophilia, on the X chromosome, and it is recessive. Since women have 2 X chromosomes, they would need to have the mutation in both of their X's in order to have the disease. Men, on the other hand, have only 1 X chromosome. So if that 1 chromosome has the mutation, they don't get a second chance. They don't have a good X chromosome to dominate over the mutant one. This explains why the disease is more prevalent in men than in women. The same applies to color blindness. It too is linked to the X chromosome and is more prevalent in men than in women. Recall that men pass their Y chromosome to their sons and their X chromosome to their daughters. So men never pass the bad gene to their sons, and they always pass the bad gene to their daughters. Women who are carriers, 1 bad gene, 1 good gene, pass the bad gene to their offspring half the time. As an example, let's look at this tree with which by now you are familiar. Queen Victoria was a carrier of hemophilia. Victoria passed the bad gene to her daughter Alice, who in turn passed it to her daughter Alexandra, Tsarina of Russia. And Alexandra then passed it to her son Alexei, giving him the full-blown disease, not just the carrier for the disease. But Victoria did not pass it to her son Edward, King of England, so Victoria kept her own house clean. Victoria also passed it to her daughter Beatrice, thereby infecting the Spanish royal family. And she also infected the Prussian royal family. So she infected 3 royal families, Prussian, Russian, Spanish, but she kept her own house clean. That brings us to the end of my presentation. Thank you all for attending. I know that you've been a wonderful audience, even though I can't see you or hear you. We've covered a lot of material, and I'm sure you weren't able to absorb it all. I do have a paper based on this lecture, and you can read that to help you understand the parts that you didn't follow today. That paper is on my website, stevemorse.org, and is in the publications section. The title of that paper is the same as the title of today's talk.
Okay, well, that gets us to the end of the lecture. I've received several questions in advance, so let me go over some of them and give you the answers. A very common question is, which DNA test would you recommend? As I mentioned at the start of the talk, there are 3 different types of tests. One that tests your paternal line, the Y chromosome test, one for your maternal line, which is the mitochondrial test, and one for all lines. The test for all lines is the so-called autosomal test. The Y and mitochondrial test are based on science and therefore are the most reliable. The autosomal test is based on statistics and could give many false results, positives as well as negatives. Also, the Y test is for both early migration pattern, the Snips, as well as for finding close cousins, the STIRs. The mitochondrial test is for early migration only, although in exceptional cases you can use it for finding close cousins. That was done in the case of Anastasia, where there were some very rare mutations found. Recommendation: the Y chromosome is best. The mitochondrial test only if you want to learn about your very distant female cousins. The autosomal test is the least reliable, but is the only test that covers all your lines. Caveat: all of the major testing laboratories except one has stopped doing the Y and the mitochondrial test. So if you want the Y test, your choices are somewhat limited. Next question: which testing company do you recommend? Well, I'll refrain from answering that because I want to maintain neutrality on the subject. But if you want to do a Y chromosome test, you don't have much of a choice. If you want an autosomal test, you probably want to pick the company that has the largest number of samples. Next question: is it worthwhile to submit DNA to more than one company? Well, some companies will import your results from other companies. This would be less expensive than doing a test from scratch at that company. So rather than submitting to 2 different companies, check to see if one of them lets you import your results from the other. Next question: is it possible for a daughter to trace her father's genealogy when there are no brothers or male cousins? And I assume this person also means no paternal uncles as well. Well, unfortunately, this person is out of luck as far as doing a Y test. I had the same situation in my own testing. I discovered a woman with whom I wanted to compare Y DNA results with, but she had no males in her family to test. Her father was deceased, so they did have a sample of his DNA from a biopsy that had been preserved. I asked Bennett Greenspan, the head of one of the testing companies, if they could accept that. He replied no, because he said it would be too unreliable. Next, can you explain why Y chromosome DNA is passed down to people who are apparently unrelated and don't even have the same surname? Well, they are unrelated only as far as you know, but you don't know about the non-paternal events in your family, adoptions, extramarital affairs, etc. That would explain the common Y chromosome and would also explain the different surnames. Question: I have thousands of autosomal matches, but I have not been able to match any of those matches to my family tree. Answer: as I said, the autosomal test is based on statistics and not science. It is possible to get false positives due to where the chromosome splits when 2 chromosomes are being shuffled together. That split is random for each birth and would affect the common length of the DNA being passed down. Question: why is my ethnicity percentage different between the different companies and even different at different times from the same company? Ah yes, the ethnicity tests. There are 2 uses made of autosomal testing. One is for finding matches, and although that is less reliable than the Y chromosome test, it does sometimes give remarkably good results. The other use of autosomal testing is for obtaining ethnicity results. The ethnicity test is the least reliable of any of the tests. It is based on statistics, so that's already one strike against it. But worse yet, it assumes that we know what the DNA sequence is of the standard person of that ethnicity. Who was the standard person and how did they identify him? I constantly tell people that the ethnicity results makes for wonderful parlor conversation, and I know I'll get a lot of flack for saying that. But you shouldn't put too much weight on your ethnicity results, but rather take it with a grain of salt. Question: what is Jewish DNA? What markers are they looking at? Is there a certain DNA sequence that is associated with Jewish lineage? Well, that gets into the ethnicity testing. They are not testing for a specific gene for Jewishness. Instead, they are comparing your DNA to what they believe the DNA is for a standard Jew, what the DNA for a standard Jew should be. How they determine that standard Jew is questionable. Question: I live in Israel. What DNA tests can I do? Well, Quite honestly, I didn't know that DNA tests were restricted in Israel until I started reading all the questions that I just received. Apparently, the Israeli government has put severe restrictions on who can get a DNA test. I don't know much about that other than what Google has to say, so I suggest you just Google it. Question, what about endogamy? Well, obviously don't do it. Endogamy is a result of inbreeding and results in your having the same ancestor on more than one line in your tree. But if you happen to be in a group in which endogamy is common, such as Ashkenazi Jews, you will get matches that appear to be closer than they really are. That's because you will match that person from more than one line in your tree. You can consider this as a good thing because it allows you to find a match whom you otherwise might have overlooked, but it makes it all more challenging to try and figure out which line in your tree that match is on. Question, could you illustrate how matching segments distinguish between first and third cousin relationships? Illustrations would be helpful. Well, this is the realm of autosomal testing. It was not covered in this talk. However, I do have a separate talk and a paper that deals with autosomal testing and gives the illustrations you are asking for. Just like this talk, that paper is also found in the publication section of my website. It's titled DNA Beyond the Y Chromosome, Autosomes Exposed. Question, what about privacy issues? What happens if my testing company is sold and data winds up in the hands of a health insurance company? Well, a lot of people are apparently very concerned about privacy. If that's you, my best advice is to not get tested. Also, don't allow any of your cousins to get tested either, because you've all heard about the famous murder case that was solved because of a match to the DNA of the murderer's cousin. You've got to have some level of trust with the testing companies here. Will the testing company divulge your DNA to law enforcement officials? Will they themselves use it for nefarious purposes? This is before you even worry about the company being sold in the future. So, again, it's a matter of trust. Okay, those are the most significant questions that I've gotten in advance. Rami, you might have some questions that came in during the lecture.
Well, there were a lot of questions that came in, and thank you for the amazing presentation. Perhaps we could just talk a little bit about the Kohan gene. There are a number of questions that came in about that.
Yes, the Kohan modal haplotype. This is one that was spoken of a lot in recent years. You don't hear much about it more recently, and that's because I think it's flawed. The test itself is based on 6 markers, and they go back to all the descendants of Aaron, which are the Kohans, which goes back 3,400 years. Now, from what I've read, 6 markers is not sufficient to go back 3,400 years. There is a 12-marker test that they're using. And they've located down to 12 markers, but that test has never been peer-reviewed. It's never been published, and it won't be because the company, that testing company that came up with that test, is keeping their results, keeping it proprietary. So without that test being published, we're down to 6 markers, and as I said, that doesn't go back far enough. Now, there are many anecdotal evidence in my own family, my own people I know who are certain about being Kohanim, and the tests indicate that they're not. In fact, I have 3 such examples. I won't mention them, but 3 out of 3 cases in my own family in which the Kohanim test gave very results which were not expected at all. That's a small sample size, but yet it's 3 for 3. Also, there are many non-Jews who are coming up with the Kohan modal haplotype. And there was another point, and I can't think of it now, regarding the Kohan modal haplotype. Oh, yes. The testing companies, in response to the criticism about the CMH being so unreliable, giving such strange results, they came out with a statement that perhaps they got a little wrong there. There's more than one Kohan modal haplotype. And I scratched my head and I said, what does that mean? Did Aaron have more than one set of DNA sequences? That doesn't make sense. So again, I'm going to get a lot of flack for saying this, but I don't give much weight to the Kohan modal haplotype test.
Okay, thank you very much. We're going to wrap up the presentation. I'd like to thank you again, Dr. Morris, for this program, for this webinar, which was very informative. We received a lot of very positive feedback already. For everybody who is watching, a recording of this presentation will be available on our YouTube channel, and we will also be distributing the handout that Dr. Morris prepared. That's the 29-page guide. We will include the link to our YouTube channel in the email that goes out this afternoon. I'd like to remind everybody that we have one more Jewish Gen Talk taking place next week with Dr. Sally Ann Sachs. She'll be talking about Jewish names. You can register for that by visiting JewishGen.org/live. We thank you all again for joining us. Dr. Morris, thank you again. Wishing you all a wonderful day.
Thank you for inviting me.